here are some basics. DNA is made up of a bunch of nucleotides, which are made up of sugar, phosphate, and one of 4 nitrogenous bases. These are their chemical structures, but that's not completely important right now. In the animation, black is used as the backbone, which is made up of phosphate and sugar, and the bases correspond with these colors, key listed here. Adenine and thymine bond together with 2 hydrogen bonds, which isn't shown in the animation because that's tedious, but I'm pretty sure there is one frame showing hydrogen bonds. Also, guanine and cytosine bond together with 3 hydrogen bonds. And that's what you missed on bio. Alright, so what you just saw was DNA replication. We start off with the cell, which is where the nucleus is located. Furthermore, DNA is located inside the nucleus. Here we can see this purple-pink circle and the DNA strand interact. That circle is an enzyme known as helicase, which acts as a zipper and untangles the DNA strands. This is where it gets complicated, so I'm going to slow down and zoom in different parts of this process to go more in depth. Let's start with the leading strand. The leading strand in this case is the bottom strand. The end we see here is the 3' end, and since DNA replicates from a 5' to 3' direction, it's really simple. The light blue oval is an enzyme known as DNA polymerase, and it starts the parent's leading strand and goes through, synthesizing the other DNA strand that complements it. Let me go back a minute to the first nucleotide it creates. As we can see, the polymerase made a mistake in matching base pairs. In this case, the polymerase simply went back and proofread, fixing its mistake. From there on out, it's smooth sailing, and it's simple. It just goes through and creates the other DNA strand. So let's move the attention onto this top strand being replicated. Then we see here is the 5' end, and since, as I said, DNA is only replicated 5' to 3', something else happens here. We can see that once the helicase gets to a certain point, another DNA polymerase comes and creates new nucleotides. However, once it gets to the end, it has to stop. There's no more nucleotides to replicate. By then, helicase has continued to separate the DNA, so the polymerase can come once again to create new nucleotides and stops when it hits the other section. These broken sections are known as Okazaki fragments. You might have noticed that a gray circle came in and connected these Okazaki fragments. In order to create a coherent DNA strand, the enzyme ligase comes and glues it together, and it just goes through this until there's no more DNA to replicate. And voila, 2 new DNA strands made from 1 parent strand. Here's the animation again, now that everything has been explained in depth. Okay, before we start protein synthesis, let me point out some key differences. While DNA is made up of 2 strands, RNA is only made up of 1. That's a huge structural difference. Another huge difference is that in RNA, thymine is not one of the nitrogenous bases. Instead, it's replaced by uracil. Here is its chemical structure. Another difference is that RNA has ribose as its sugar instead of the deoxyribose sugar that DNA has. Here are its chemical structures. You can see the difference highlighted in blue. There are 2 parts of protein synthesis, transcription and translation. Transcription occurs in the nucleus since that's where DNA occurs, and translation occurs in the cytoplasm. More on both of these concepts later. Yeah, I'm not going to make a Glee reference this time. see, protein synthesis is a little more complicated. Again, I'm going to zoom in and out and slow things down as I explain everything that's happening here. We start out inside the nucleus, where we see our DNA strand interacting with a dark purple oval. This oval, called RNA polymerase, essentially splits the DNA strands for a very short amount of time to create an mRNA transcript. If you look closely, you can see that the mRNA transcript is made from the leading strand of the DNA strand, using the complementary base pairs. As I mentioned earlier, thymine is not a base in RNA, so whenever it needs the base pair of adenine, the mRNA transcript has uracil. This transcription is essentially making the mRNA. As we move on to the translation portion of protein synthesis, we see that the mRNA strand is leaving the nucleus, or in this picture, the golden circle. It makes its way over to this lighter purple circle, a ribosome in the cytoplasm. As I said earlier, translation occurs in the cytoplasm. We watch this mRNA strand attach to the ribosome, becoming rRNA, so translation can really start happening. Then a tRNA comes down. Let me focus on these bases really quick. A group of 3 bases in rRNA is called a codon. The tRNA has the anticodon, the complementary respective base pairs, and on the other end of that is the amino acid that corresponds to this codon-anticodon, according to the universal genetic code. Notice how the tRNAs don't start right away. Instead, translation always starts with a codon that reads AUG. This is the start codon that corresponds with the amino acid abbreviated as MET. This repeats until there are either no more codons or until it reaches a stop codon. In this case, we simply ran out. The amino acids that connected on the top form a polypeptide chain with polypeptide bonds. This is a protein, and this is how proteins are made. Going back to our cellular function unit, this makes sense because we defined the ribosome's purpose as making proteins. This is a more in-depth explanation of how that actually happens. This process is very important considering how proteins are necessary for almost anything we do. Really quickly, I'm going to show an analogy on the video in words to help understand this process better, inspired by the baker's analogy.